This video is talking about osmosis and dialysis. With these two, we are going to discuss the permeability of the cell membrane and then determine what happens with different solutions. So the cell membrane is a selectively permeable membrane that allows water and certain solutes to cross the membrane. Colloids and larger particles cannot cross this membrane. We will also see that ions struggle to cross the membrane because they have a charge associated with them, that neutral molecules do better here. So a very important vocab word is diffusion. And what this is the random movement of solute particles through the solvent. This random movement is obtained through the kinetic energy of the particles. So this comes from the kinetic energy of the particles. Diffusion results in the uniform concentration of the solutes. So it makes everything even throughout. And we will see that that is called an isotonic solution. Two solutions of different concentrations separated by a selectively permeable membrane creates a concentration gradient. There will be more to come with that on the next couple slides. So permeable substances, permeable means it's allowed to pass through, it can pass through. Readily diffuse. What can readily diffuse? Small molecules. You will see that it does not matter if it is nonpolar or polar. Small molecules can readily diffuse through a cell membrane or through any membrane. Stuff that is permeable but needs some assistance are larger polar molecules like glucose here. We'll see this one quite a bit. And small monatomic ions. So Cl minus, Na plus, K plus. Your less permeable substances. The least permeable ones are large polyatomic ions. So if you're thinking of phosphate, for example, any colloid will not be able to pass through a membrane. And impermeable, that it just won't happen no matter what. You have your proteins, your polysaccharides, and your fats. These are all very big molecules. So your factors that really determine whether a solute can cross a cell membrane, there's three of them. Solute size, smaller is better. Solute charge, we want neutral. Let's write them down. So small is good, neutral is good, and the last one, solute polarity. Less polar is better. So nonpolar is better than polar here. So here's a table showing three different electrolytes and then the concentrations inside the cell versus outside the cell. So because the ions cannot diffuse across the cell membrane, remember we want neutral things, there are something called ion concentration gradients. Ion concentration gradients. And this is between the inside and the outside of the cell. These are very important because these guys help with the transmission of nerve impulses. That along with other signaling in the body. But these ion concentration gradients are very important to have. Just something to point out here, the units, lowercase m, capital M, that stands for millimoles per liter. Which ion or molecule in each pair is more permeable and better able to cross the cell membrane? So the first group we have here, we have an ion, which is chloride, or a neutral molecule, which is glucose. So glucose will be better there because it is neutral. In B, we have glucose or water. They're both neutral. There's no charge there, but water is much smaller than glucose. And then we have oxygen or potassium. Again, potassium ion, that is an ion that has a charge to it. Oxygen is neutral, so that will be better. Also, something to point out for oxygen, it's nonpolar. So that helps it as well. So here we said glucose because it's neutral, water is smaller, oxygen is neutral and nonpolar. Okay, here is some more here. So we have calcium ion or a small neutral amino acid molecule. Well, small and neutral are the big parts there. We have water or sodium. Water, again, it is neutral. Carbon dioxide or potassium ion. Neutral. It also happens to be nonpolar. And then we have starch, which is a colloid, or glucose, which is small. It says they're both neutral. Again, glucose, the small part helps. Now, the section does focus in on osmosis and dialysis. So here is osmosis. Osmosis is the diffusion of water across a selectively permeable membrane from the less concentrated to the more concentrated. We will see that dialysis is the opposite of this. So we have to keep them separate. Osmosis is from less to more. So when it goes from less to more, you can see that the volume of the more will increase, which should make sense there. That water is going to go to the more, meaning the more has more volume. So let's look at this diagram here. So on the left here, we have before osmosis. And we can see that the volume of them straight across is the same, right? Same level. But notice your little black dots here, those are your solute particles. The seawater has a lot more of those particles, of those solute particles. The freshwater only has a couple there. So what's going to happen, that the less concentrated will flow to the more concentrated. So these guys are going to cross this membrane, making this level, not those, the water itself is going to cross here, making the level of the seawater go up. And that's what you can see on the right side here, that after osmosis, look how much higher that volume is. So that is what osmosis is. Now, something called osmotic pressure. That is external pressure that must be applied to the more concentrated solution to prevent osmosis. So what would happen here, we would need some kind of pressure coming down here in some form to prevent osmosis from happening. And then if that pressure is greater, if the external pressure is greater than the osmotic pressure, that would cause what's called reverse osmosis. So that's here. So reverse osmosis is when the external pressure is greater than the osmotic pressure. And you can see what happened here where this pressure was applied. The water flowed in this direction. Okay, tonicity, three very important vocab words that we're going to use a lot. Hypertonic solution. That is more concentrated. Hyper, more. Hypotonic with an O. That is less concentrated or more dilute. That will have less solute particles. And then isotonic solution, that is equal concentration. So if we're looking back here, you're more concentrated. This is called hypertonic. The less is called the hypotonic. And then when they're equal, after osmosis occurs, that's B here, they are both isotonic. Same concentration. So this is pretty interesting, what happens with red blood cells. So if we look our extracellular solution, this is outside the cell, and then intracellular, this is inside the cell. So when these red blood cells are immersed, you can see right now in A, it's an isotonic solution, that the concentration is the same. And you can count your little dots here. These are your particles, they're 6. So isotonics are the same. In an isotonic solution, you will have no net flow of water. Nothing needs to happen. In B here, what's happening in the hypotonic solution is that your water flows into the red blood cells. And what's happening now, what causes that to happen? So it's flowing into the cell, it causes the red blood cell size to increase. It's absorbing water, and then they may burst actually. And that's something called hemolysis. And you can see what happened here. It got enlarged. So it went from the outside of the cell into the cell. And then C, sorry about that. C, our hypertonic solution. This is when water flows out of the cell. And that causes the red blood cell size to decrease, or it will shrink. And that's something called crenation. So very important points there. Again, this is from inside, and then the water is leaving the cell, and you can see the cell is shrinking there. And this is all because the solute particles, the water is flowing from the less concentrated to the more concentrated. So here, again, is the same idea, before osmosis versus after osmosis. And we're going to have questions on the next couple slides. So if you want to pause them before I give you the answer, that would be a good thing to do. So it says, consider aqueous solutions A and B with different concentrations and separated by a selectively permeable membrane. The small circles represent solutes that cannot diffuse across the membrane. So it says, identify the hypertonic and hypotonic solution before osmosis. So if you're looking at your solute particles in A versus B, there are less in A than there are B. So A would be your hypotonic, and B would be your hypertonic. Step B now, it says after osmosis has occurred, label them as hypertonic, hypotonic, or isotonic. After osmosis, the concentrations are equal because that water will only keep diffusing until it's equal. So now these are isotonic, the same concentration in A and B. Of course, we can see that the volume of B has risen because it flowed from A to B. Step C, I just answered that. Explain any changes in volume. Again, it's because the water flowed this way that it caused the volume in B to increase. So it's just the way the water diffused. And D says, to which solution before osmosis would you apply pressure to prevent osmosis? So remember, we are applying that to the hypertonic solution. So you would want to apply pressure here. So B, because the water diffuses or flows from A to B. So whatever is your hypertonic, that's where you want to apply the pressure. And that would cause reverse osmosis, which would then make the flow go this way. What happens when a red blood cell is placed in a 1.5% solution of sodium chloride? So you need to know that a red blood cell is 0.90% mass volume sodium chloride. So if you're looking at the 1.5%, that's obviously greater than the 0.9. So that 1.5% is your hypertonic. This would then be your hypotonic. So the water will diffuse out of the red blood cells because it's going from your hypo to your hyper, resulting in crenation. Okay, so that was osmosis. Now, dialysis. This is the opposite of osmosis. This is from hyper to hypo. So you're going from greater to less, a more concentrated to a less concentrated. Here, notice you went from a less to a more. Here you're going from more to less. So opposite. You're still going to have your small solute particles. That does not change. So what can go through the semipermeable membrane remains the same. So here we have an example. It says mixture A contains urea, which is the little blue, small blue particles. Albumin, which is the larger yellow ones. It says that's a colloid. Describe what the two compartments will look like after dialysis. So what's going to happen, only the small ones can pass through. So the urea will diffuse from hypertonic to hypotonic until both concentrations are equal. And then when they're both equal, we know that that means they will be isotonic. So these urea molecules will go over here. Only the blue ones, though. I think that says this on the next slide. Yep. So your urea will cross the membrane. The yellow, the colloids will not. So if you had to draw this, you would just draw it where the blues, you would have about the same on both sides. And then the yellows wouldn't change. And it would look something like that. There it is. Okay, and finishing up here, it says consider two solutions that are separated by a dialysis membrane. One side contains 0.45% saline and the other contains 0.9%. Which solution is hypertonic? Hyper, this is the more concentrated, so this would have to be the 0.9%. Hypo is less concentrated, which would just be the other one. In which direction will the sodium and chloride ions diffuse? Because we are talking dialysis, we're going from hyper to hypo. So this would be the 0.9 to the 0.45. And then how will the concentrations of each solution change? Well, the 0.9% will go down, and the 0.45% will increase.